The mechanisms for cellular transport can be categorized into three main groups. And the first one is going to be passive transport. Passive transport has two key characteristics that I want you to remember. The first one is going to be that in passive transport, materials are going to be moving from high concentration to low concentration. This is what we mean by down a concentration gradient. So if I can circle this, this is going to be the first main characteristic for passive transport. And the second one is going to be that passive transport does not require the cell to use additional energy other than the free energy of the particles that are constantly moving in solution. So the cell doesn't have to use energy. One of the most common forms of passive transport, and will help me illustrate this concept, is known as diffusion, just simple diffusion. The illustration that I'm using on this slide can be practical for this. You can see, for example, particles or molecules of dye, green dye, that are mostly concentrated on one side of what is represented here as a plasma membrane. And so these gray dashed lines you see over here, that is what represents a living plasma membrane. And what one can predict, if we could imagine these green solute molecules of dye as they're moving and colliding with one another, they're going to be going from a region where they're more concentrated to a region where they're going to be less concentrated. And that is what we see as we go to the illustration in the middle. Some of these green solute particles have been traveling from where they are more concentrated to where they are less concentrated, and eventually they reach a state known as equilibrium. At equilibrium, one has to recognize the following. When a solution is at equilibrium, it doesn't mean that the solutes, the particles that are dissolved in solution, are not going to stop moving. When a solution is at equilibrium, it simply means that for every particle going from the left to the right, as I'm using this illustration, there's going to be another one going from the right to the left. This is also known as dynamic equilibrium. And that is what happens in a cell if equilibrium happens to be reached. In most cases, cells are not going to quite achieve this level of equilibrium because the movement is going to always be leading to shifts, changes in the concentration of solutes in the solutions that are important to the cell. So this is a good example for passive transport because particles are going from where they are highly concentrated to where they are less concentrated, and you don't see here any reference to the cell having to use energy. Another example of passive transport is going to be osmosis. Osmosis, some people define it as the diffusion of water, but I like to be more precise. I like to say that this is the movement of water from regions of higher water concentration to a region or a solution that has a lower water concentration. And so, once again, I have illustrations that are going to be helpful in making this point. As you look at this U-shaped tube, you're going to see that there are two solutions that are separated by a selectively permeable membrane. What is allowed to go from one side to the other is going to be the water, represented here as light blue. But there's going to be purple solute particles that cannot move through this plasma membrane. You may be asked the question, what would be the nature of the movement of water? Well, if you look at this side over here, you can almost get evidence that the water is more concentrated because there is a greater area that is occupied by this light blue. And so water is going to be more concentrated on the left side because there's less solute. And so there's always this trade-off. If you have greater solute, the concentration of water is less. If you have lower solute, the concentration is going to be greater if you're comparing two solutions. So water here is going to be moving from the left to the right, or from a region of higher water concentration to a region of lower water concentration. And so you see, here we have again that concept that in passive transport, particles in solution are going from high to low concentration. Analogies are sometimes helpful. In terms of energy, imagine you are on a bicycle at the top of a hill, then there's this top of the hill, and there is a nice paved road with a gentle slope to take you from the top to the bottom. All you have to do is keep your balance, but you don't have to pedal. You don't have to use additional energy other than keeping your balance. And you can go from the top of the hill all the way down to the bottom without any additional use of energy. So that's the idea of going from high to low concentration cells. Don't have to use more energy. And so what happens here is that water continues to move, and as it goes from the left to the right, notice how the right-hand side, the level of this solution is going to go up. Because the solute particles are not moving back in this direction, there's going to be an uneven level for the solution. Now in this window, you can see like a closer look at what is happening. The solute particles are being surrounded by water. This is what we call a hydration shell. And this makes less free water on this side and makes it possible for water to be moving from where it's more concentrated to where it's less concentrated. I'm sorry, my computer is slowing down here. Okay, and whenever we're talking about osmosis, we are going to always have to identify three possible scenarios. One is going to be that a solution has a greater solute concentration compared to another. The solution with a greater solute concentration is going to be described always as hypertonic. The solution that has a lower solute concentration is going to be known as hypotonic. And the solution that is going to be equal to another in terms of the concentration, those are going to be known as isotonic solutions. And so maybe one of the things that will be helpful to you at this point is if we can recognize what are the components of a solution. So a solution is always going to be the homogeneous mixture of solute and solvent. In biology, the universal solvent is going to be water. And the parts that are dissolved in water, like water in the cytosol, the liquid part of the cytoplasm, water is going to be dissolving carbohydrates, it's going to be dissolving amino acids, it's going to be dissolving nucleic acids. A number of molecules that are important to the cell will be dissolved in water. So we have three concepts here to consider: hypertonic, hypotonic, isotonic. These are terms that are used to describe the tonicity or the relative solute concentration of a solution. Solute, not water. And so let's keep this in mind as we look at the possible outcomes to cells that are exposed to various kinds of environments. A cell in a hypotonic solution means that the cell has greater solute and the outside solution is going to have a lower solute concentration, or let's say less solute. And what happens in this situation is that water is going to be moving from where you have a greater water concentration. Greater water is expected to be found where you have a lower solute concentration, and water moves into the cell. In a hypotonic solution, a cell can gain so much water. In the case of an animal cell, which is only surrounded by a flexible plasma membrane, this cell is going to lyse, is going to rupture. The plasma membrane will break because it will not be able to hold the contents of the cell. Plant cells, on the other hand, can take a lot more water, and the reason is that they can expand a little bit in volume, but not too much, because in addition to the plasma membrane, they also have this thick and rigid cell wall. So if you were to ask a question, it's a hypotonic solution, like I'll tell you what's hypotonic: pure water. Pure water is hypotonic compared to the inside of any cell. In this hypotonic environment, who gains? Animal cells don't survive there. Animal cells are going to lyse because they are taking up too much water. Plant cells don't lyse. They are going to achieve internal pressure, which is ideal to a plant cell. This is known as turgor pressure. We can say that the cell is turgid, and this is going to be ideal to a plant cell. And so as I go back to my previous slide, I can say that a hypotonic environment is going to be ideal for plant cells, but not for animal cells. Animal cells are going to lyse. They're going to die in a hypotonic environment. What if we look at a hypertonic environment? Now let's go to the other side of this situation. In a hypertonic environment, now means that the outside has greater my writing is not working here very well. I want to erase this. There we go. And so now the outside has a greater solute concentration. Greater solute means lower water. And keep in mind, water will move by osmosis to where you have the lower water concentration. Inside of the cell is going to be a lower solute concentration. And for that reason, that really is not very legible. I'm just gonna erase it. The opposite of what is on the outside is going to be on the inside. So water is going to be, maybe I can do that. Maybe I can do greater H2O. There's greater water concentration, so water goes from inside of the cell to the outside, and these animal cells are going to shrivel. Not good for an animal cell. That cell is going to die. And if you look at plant cells are also going to be suffering because they are losing most of their water, losing most of their volume. The plant cell is said to plasmolyze. And the plasmolysis of a plant cell, again, leads to the death of the cell. So if we go back and we take a look at these situations, we have a hypertonic environment. It's detrimental. It's going to be harmful to both plant and animal cells. So as we look at an account here, hypotonic, it was good for plant cells, but bad for animal cells. Hypertonic is going to be harmful to both plant and animal cells. So what's good for an animal cell, you may be asking at this point. And it's going to be the situation we have here in the middle, an isotonic solution. In an isotonic solution means that the concentration of water in solutes is the same inside as it is outside of the cell. Now we are dealing with a situation like what we described before, equilibrium, a dynamic equilibrium. For every water molecule that moves into the cell, there's going to be another water molecule that moves out, but overall, the volume of the cell doesn't change. Animal cells love this kind of an environment. Plant cells, not so much. Because remember I said that plant cells like to have this internal pressure created by all the water that they can have. Plant cells use water, for example, for photosynthesis or for dissolving other solutes they may need to move or store, depending on the function of the cell. So in this situation where water moves in at the same rate as water moves out, the plant cell never develops the internal turgor pressure they like to have. Instead of being turgid, this cell is going to be flaccid, and that is not ideal for a plant cell. So finally, we can say that in an isotonic environment, yes, animal cells will thrive. This is ideal for animal cells. apologize for my technical difficulties. But I don't give up. I'm a persistent person, and I hope you are too. Okay, so now we've covered the effects of osmosis on plant cells and animal cells depending on the environment where they can be found. A hypotonic environment, an isotonic environment, or a hypertonic environment. And try to remember which one of these will be ideal for the animal cell, or which one is ideal for the plant cell, and which one is detrimental to both. One last case of passive transport, and I think I'm going to be stopping this video, maybe making another one for more membrane transport. It's facilitated diffusion. Facilitated diffusion makes use of transport protein. So this is most likely going to be a channel protein, one that doesn't require the cell to use energy, because remember, we're talking about passive transport. One of the conditions of passive transport is that the cell doesn't have to use energy. So this channel protein, which is a transport protein, can allow for solutes like sugars to move in. This transport protein may be one like an aquaporin that allows for water molecules to go from one side of the cell to another. And so these channel proteins can be used for moving things like substances that are going to be dissolved in water, but otherwise cannot make it through across the phospholipid bilayer. I will make another video to explain the other forms of membrane transport, which are going to be active transport and bulk transport.